Hello everyone, I'm Sankalpa Chakraborty, a first-year graduate student in Molecular Biosciences graduate program. I'm working under the supervision of Dr. Viswanathan Rajagopalan, and today I will be presenting our research on aberrant cardiac long non-coding RNA profile following altered thyroid hormone levels. Here is our poster. For better visibility and clarity, I will break it down into its major components. Cardiovascular disorders are number one killer not only in Arkansas and the entire United States, but also in the world overall. With 40% of the ongoing coronavirus COVID-19 pandemic patients dying with cardiovascular complications, our research becomes highly crucial and urgent in these unprecedented times. Abnormalities in thyroid hormones can adversely cause or contribute to cardiovascular diseases. Thyroid hormones are the master controller of the cardiovascular performance. There are two types of thyroid hormones, T3 and T4, both released from the thyroid gland. Dr. Raj Lab has published multiple papers showing beneficial effects of thyroid hormones in cardiovascular diseases. We are now on to discovering even some better therapeutics. Before going to the RNAs, the central dogma of biology states that DNA, which consists of innumerable genes, produces RNA, which in turn produces protein. But if we code all the proteins back to the DNA, it covers less than 5% of the total DNA size. So it becomes crucial to study the less explored non-coding regions. We call these RNAs non-coding RNAs. One such variety is called long non-coding RNAs, which are relatively new and emerging molecules to explore in cardiovascular biology, as evident in the picture. They comprise of at least 200 nucleotides, which in turn are the building blocks of DNA and RNA. Our hypothesis is that abnormal alterations in thyroid hormone levels will impair long non-coding RNA gene expression in the heart. The various techniques expanded here include gravimetry, ultrasound ECG scan of heart, as well as qPCR for long non-coding RNA expression and ELISA for thyroid hormone level, both of which are used to detect coronavirus COVID-19 patients. We also did exosome isolation and Western blotting, as well as statistical analysis. We first established hypothyroidism or low hormone levels and hyperthyroidism or high hormone levels in mice using excessive amounts of compounds called PTU and T4 respectively. This slide demonstrates that with hypothyroidism, the heart and its components reduced in size, and with hyperthyroidism, they increased in size. As is tested for all heart patients, including those with coronavirus COVID-19, hypothyroidism reduced heart function and decreased heart rates. Hyperthyroidism, on the contrary, showed opposite functionalities. Importantly, RNA purified from the heart tissues of these animals show that long non-coding RNAs are significantly impaired in both hypo and hyperthyroidism, especially in a differential and opposing manner. Since detection of long non-coding RNAs in blood can serve as an excellent diagnostic tool, we isolated exosomal vesicles, which are major carriers of the long non-coding RNAs in blood serum. As a proof of principle, I was successfully able to isolate and then confirm the enrichment of exosomes through Western blotting technique using CD63 antibody. In summary, this is the first work of its kind detecting heart-specific long non-coding RNAs following altered thyroid hormone levels. We strongly believe that these findings, along with the future expansion of exosomal studies, will enable us to discover novel diagnostic and therapeutic strategies for not only coronavirus COVID-19 patients, but also all patients with cardiovascular disorders. Following are the references that we have used for this presentation. We would like to acknowledge and thank Arkansas State University, Arkansas Biosciences Institute, and NYIT College of Osteopathic Medicine for their support. Last but not the least, thank you so much for your attention.